name is Mick Finn-Levere. Welcome to another ALS Expert Talk Series. So glad you all could spend your time with us because we feel that you're an extended family and we're here for you. I just want to remind you that we have a library full of all of these presentations. So if there's any time that you couldn't make it, just go to our website, everythingals.org/events, and check out the videos. Thank you again for spending your time with us. I know it's valuable and we treasure it. Okay, over to you, Indu.
So thank you. We have a special guest today. We want to welcome Dr. Jeffrey Rosenfeld, who's going to give us the topic of updates in ALS MND, the challenges and opportunities for 2023. And it's our honor to have Jeffrey Rosenfeld, who is a professor of neurology, as well as associate chairman of the department and director of Neuromuscular ALS MND Program at Loma Linda University School of Medicine since June of 2015. Dr. Rosenfeld has established the Center for Restorative Neurology at Loma Linda University, where he also serves as a medical director. And formerly, Dr. Rosenfeld was Chief of Neurology at University of California, San Francisco, which is SF Fresno, and Director of Neuromuscular and ALS Program at UCSF Fresno, has over 25 years of experience treating neuromuscular patients with extensive efforts in ALS. And so with that, I would like to welcome Dr. Jeffrey Rosenfeld, and we're really honored to have you here. And please give your presentation. And after your presentation, we have our fellows, our student fellows, who are going to do the moderating with the Q&A on the chat. So you don't have to worry about the Q&A on the chat. Thank you very much.
All right. Thank you very much, Indu. Hopefully you can all hear me okay.
Yes.
It's working very good. It's really my pleasure to be here, and I had in the past had one other opportunity to present part of a talk to this group some time ago now, and I was just amazed about how much feedback I got, even weeks to months afterwards. So it was my pleasure to accept another invitation. I'm going to try to share my screen here. Hopefully this will work, and I'm hoping that you can now see the title slide. Very good.
Yes.
So I guess some of the names and the folks I'm seeing online here are folks that come to our center. So welcome. Very nice to see you from all over. So this has really been an incredible year, an incredible time for ALS, motor neuron disease. And I think by anybody's account, this year has been noteworthy. We now have, as most of you know, 4 approved therapies for ALS, motor neuron disease. I can tell you in almost 30 years of practice, I have never said that sentence before. I have never said that there are 4 therapies FDA approved. Now, that's good news. And there's nothing that I want to say tonight that would detract from the fact that that is wonderful, that we now have more approved therapies than ever before. However, what I do want to talk about tonight is, if I can get this to go here, let me just move my slide here. What I do want to talk about tonight is a bit of an update about where we are, because as good a news as that is, I think I want to highlight for you a little bit of uncertainty that I have, a little bit of a concern maybe, as well as excitement about what's on the pipeline. And so this is going to be a little bit of behind the curtain of what it's like in the research side of studying ALS, motor neuron disease, and how we got where we are. So I thought I'd break up these comments tonight into 3 areas: where we were, where we are, and where we're going, and talk about them in that order. To start off with, the field of motor neuron disease has been an active area of investigation for, I don't know, many, many years, certainly long before I got involved. But it really has picked up momentum. And this is a slide that my colleague and friend Matt Harms from Columbia presented recently at a meeting, and I borrowed from him because it shows how many articles were published each year since 1980. So for the last 43 years, it shows the rapid rate of increase. Now, if you add up all of these articles cumulatively, you get a sense, and you did the math, that is about 2 articles every day have come out on average since 1980. If you divide all of these, at least 2, perhaps more than 2 articles every day, Saturday, Sunday, holidays, that's an amazing rate of discovery. Now, if I look at this graph, when I looked at this graph, you kind of see that it's not an even linear increase in the publication rate. In fact, I look at it like this, where you can see there was sort of a plateau period in the 80s up to the 90s, and then it escalated a little bit until about the 25, and then it went up again, and it really shot up at around 2014 or slightly before. And it turns out that these inflection points on the growth of the publication rate were coincident with very seminal discoveries in ALS. And if you look here at these points, they correspond in time to these 3 discoveries. In the early mid-90s, we discovered this protein called SOD1, superoxide dismutase type 1, and found that mutations in this protein might have a lot to do with, and even in some cases cause, some forms of motor neuron disease. And then in the early 2000s, some 12 or more years after, the discovery of TDP-43, which is a different protein with a different function, seemed to be very seminal in understanding other forms of ALS. And TDP-43 has to do with routing of certain proteins in the cell nucleus. And then most recently, in about 2012, the discovery of another mutation, a genetic mutation, C9orf72, was an explosion of understanding because it tied together two different areas of investigation: motor neuron disease and frontotemporal dementia, something that we didn't necessarily think had all that much to do with motor neuron disease until this link of C9, this mutation, which we knew caused frontotemporal dementia, now seemed to also cause patients to have a motor neuron disease, many or most of which also had frontotemporal dementia. So that really expanded the field quite a bit. Now, if you look at the rate of discovery in the most recent inflection upward, it's something like 8 or 9 articles per day over that period of time. So just to give you an idea, this is an extremely active area. And I didn't have the time to go and look at another field like HIV research or cancer research to look by comparison, but it's hard to imagine that the rate of discovery and the rate of publications is going to be even more so than this. This is just to give you an idea of how much information and how many papers are out there. And part of the reason for that is, over the years, there have been some excellent hypotheses about why motor neuron disease occurs. Now, lots of neurodegenerative diseases have hypotheses associated with them about why it occurs. But the thing that always struck me in motor neuron disease is the diversity and the number of hypotheses. And these aren't just ideas. These are mechanisms that honestly have quite a bit of evidence to support. And if time allowed and I was doing a talk on just any one of these, probably by the end of the talk, I could convince you that that is the mechanism to pay attention to better understand why motor neurons decline in a disease like ALS. But in fact, there isn't one of those. There's multiple areas of potential relevant understanding for why motor nerve cells die. So could it be that all of these are right? And many of the clinical trials that we're going to talk about today, many of the drugs that we've tested are relevant to one or more of these mechanisms. Yet we haven't necessarily found a drug which I would consider to be a home run, that is one to make a very, very significant and noticeable difference to you. Nonetheless, there's an incredible amount of evidence supporting one or more of these hypotheses individually. How can that be? And that's what we're going to talk about in just a moment. More so one idea that came out of that is perhaps there are several different ways to get to the same endpoints. In other words, motor neurons can die for one of various reasons. So perhaps there are multiple insults that can all relate to what's called RNA dysregulation. That's the way in which our cells handle genetic information coded by RNA. And maybe there's more than one right answer. Maybe different individuals are triggered to dysregulate the messenger RNA differently. And so there were a number of reviews and papers that talk about how there could be more than one pathophysiology or more than one reason for motor neurons to die or to trigger. Perhaps it's inflammation, perhaps it's cytotoxicity, perhaps it's glutamate, perhaps it's calcium dysregulation, and so on. And maybe as an alternative idea, there are different mechanisms that are associated with different stages of the disease. So there might be something that triggers the disease, and then something else that propagates the disease, and something else that actually causes the motor neuron to die when it gets to a certain stage. All of these are very valuable and valid hypotheses. We're just not sure which one is the accurate one. Now, what is known about ALS, if we're still in the where we were section, is that it can be very serious, and it can actually result in a mortality of 50% by 3 years. And this is a very old slide. And I don't actually think it's terribly accurate anymore, because what's more known, what's even more prevalent, is how variable the disease is. And I don't think it's fair when you look online and you say the average person will expire in 3 years. That just is not my experience, and probably is not your experience either. It's much more likely that this is what we see, that there are different rates of progression. And this is monitored by what's called the ALS score. That's the Appel ALS score that but there are many examples of this in the literature showing that patients progress at very variable rates. So although you probably can't see my pointer here, or perhaps you can, this person in the blue line at the top was fairly stable for many, many months while this study was being done, whereas somebody over here on the left dropped precipitously, coincident with a more severe and progressive disease. So why is it that some patients don't progress and some patients progress rather abruptly? Is that two separate diseases? Is it two separate mechanisms? Is there something else driving it? Not sure I'm going to be able to answer that question tonight, but I will certainly tell you that it's a very relevant fact that it is a highly variable disorder, and not everybody definitely goes down the same pathway. So I don't even quote statistics for my patients with regard to mortality, because I think it's so variable. This is a slide that is not exactly current, but it's a slide that highlights a large number of clinical trials that have been done that are relevant to one or more of those mechanisms that I talked about. And I put this slide up here, even though it's a bit depressing in the sense, because most, if not all of those, a couple of these were positive trials, but the majority, 95% or more of these trials were not positive. They were failed trials. But this is over a large number of years. And what's interesting and highlights the point that I'm making is, if you look at this, not only did we do a lot of different trials, sometimes we did the same trial 3 times. We did it 3 times for different reasons. Sometimes there were different phases of the trial. Sometimes somebody had a better idea, but it underscores the fact that the preclinical basis for these trials, that is the evidence we had before we started a trial, was so compelling in some of these that individuals kept wanting to do the same trial again. It's hard to do a clinical trial. It takes a lot of effort and a lot of resources to do a clinical trial. So you really want to feel good about the fact that you're going into it for the right reason. But if you look at this, in some of these trials, perhaps 2 and 3 times studying the same drug, sometimes by the same investigator, just underscores how the preclinical basis that a drug should work and the reality of what we've experienced in the clinical trials are really at a bit of odds with each other. So it's a little bit humbling, but at the same time, a little bit instructive to see the history of how many trials we've done. And I certainly can assure you that this is not a up-to-date list. There's been other trials. The third column would be pretty full if I actually added to trials of the last 8 to 10 years. Sometimes we classify them by what mechanism they addressed. So those mechanisms that I showed you earlier, the drugs from the previous slide, could be categorized. So we have tried to address anti-glutamate hypotheses, anti-inflammatory hypotheses, antioxidation hypotheses, neuroprotective hypotheses. Neurotropic factors were very big when we started in the 80s and 90s, and so on. And you get a sense from the middle column that most of these trials failed. So we have a mismatch of an incredible understanding of some of these mechanisms and their relevance, yet a fairly consistent failure at addressing these mechanisms when we test it in patients. That's a mismatch that those of us that work in the field are getting, unfortunately, used to. And one of the things I'm hoping to leave you with tonight is perhaps a reason why we haven't seen more positive trials from very well-intended hypotheses. This is basically a slide that says the same thing in a different way, but I like the way it was organized, and it showed each mechanism, the kind of things that we've tried. We have addressed the cytotoxic mechanism with a variety of different drugs tested, and the neuroinflammatory mechanism, and the oxidative mechanism, the apoptotic mechanism. All of these, as I say, very well understood and relevant hypotheses, but we've come up short in the clinical trial, except perhaps for the ones that we have most recently gotten approval. So where are we right now? Well, we have riluzole, and riluzole is something that most all of you are probably familiar with. This is a drug that was approved in the mid-90s. It was a well-done, large trial that looked at about 155 patients. There was actually more than one trial. They were studied for up to 18 months. And what it showed in the graph you see on the right is that the survival rate for patients taking riluzole was better than the survival rate for taking placebo. Now, in this trial, the endpoint of survival was either an individual's death or that they went on a ventilator. They counted that as the same. So it got sort of a mantra, this riluzole, that it improves your breathing or your life expectancy for up to 3 months. A little bit of a misnomer, because subsequent studies have shown that in some patients it could be much longer than 3 months, but on average, it's 3 months. So this was our first indication that there was such a thing as a disease-modifying therapy. That is, a therapy that could change slightly the course of the disease, if not noticeable to the patient, but certainly in time and statistically in a larger group make a difference. So that's riluzole, and it addresses a hypothesis called excitotoxicity, or the glutamate as a neurotransmitter being a toxic effect onto the motor cell. It reduces that toxicity. Now we have most recently edaravone. I apologize for the typo there, but edaravone, or Radicava, was the second drug. And there were many years between the discovery and the release of riluzole and edaravone. But interestingly, edaravone was used for some time before it was even tested in ALS. It was used in other countries for treatment of stroke. And the reason why it was used for treatment of stroke is that it's thought to be an antioxidant. And oxidation is a mechanism not just in ALS, but it's a mechanism for, after a stroke, how the surrounding tissue that is sort of affected by the stroke but still alive, how it is vulnerable to oxidative insult. So it was found in Japan and Korea primarily that it would be a benefit for stroke, and so it was tested in ALS. Well, it was tested in ALS several times before the trial was positive. So this is one of those examples where the same trial was done multiple times, only in this instance, edaravone was ultimately found to show some benefit. The benefit that edaravone showed was illustrated here in this graph, where the ALS-FRS, which is a functional rating scale that you might be familiar with, a questionnaire, the rate of decline that we expect in that ALS-FRS score was less. So the red line here shows a bit of a plateauing of the FRS score, and the blue line shows the placebo group that declined with time in the FRS. And the difference at the end of the trial was a difference in the slope of expected decline, and consequently, the drug was approved. Now, it was an interesting approval because the trial was not done in the US. The trial was done in Japan and I believe Korea. And the FDA accepted those data as valid for approval, and that's why we have edaravone today in multiple forms. As you know, it was initially released as an intravenous form and more recently formulated and approved as an oral form. But what we hope to do with edaravone is, again, for a completely different reason than riluzole, but slow down the progression. That is, our number 2 disease-modifying therapy had been approved. And then most recently, the drug, the combination of sodium phenylbutyrate and tersodiol or TUDCA, as it's known, that combination drug also showed a decline in the expected rate of the ALS FRS. So on this graph here, the red one is on top, that's slowed down slope, and the green one is the placebo slope. And you can see that over the course of this 6-month trial, by the time 6 months, the 2 lines diverged quite a bit. This trial is currently, there's a trial currently going on for sodium phenylbutyrate and tersodiol, a phase 3 definitive trial, but this drug got approved with the data that they had at the time of approval based upon this graph and others that supported it, showing that patients seemed to progress slower when they were on this. Now, the mechanism of sodium phenylbutyrate is hypothesized, not really positive, but it is different than the mechanism of edaravone, and it's different than the mechanism of riluzole. So the idea then comes that perhaps taking more than one, at least theoretically, might have greater benefit because you're addressing different mechanisms. That's never been tested, but in the case of edaravone, for example, all of the patients in the edaravone trial, when it was tested, happened to be on riluzole as well. And in the case of sodium phenylbutyrate, TUDCA, many of the patients were on other drugs. So it's a bit of a complicated analysis with the numbers that we have, but it is an active question if addressing more than one mechanism at a time changes things. But nonetheless, this is how we got these 3, which are now commercially available. So let's talk a bit about where we are now, beyond what we have in these drugs. Okay, so if I break this down and say, what do we know versus what do we think versus maybe what do we want to know? We certainly have an appreciation of small steps. I think it's fair to say that these are very important discoveries, and I am enthusiastic about them, and I prescribe these drugs and I offer them to my patients, and there's nothing to diminish it except to say that these are disease-modifying drugs. So that means that they're not necessarily going to make you feel better. You may do better. You may progress in a more favorable fashion or slower fashion, but you're not necessarily going to feel that difference, largely because you won't know what it would have been like to not be on the disease-modifying drug, whether you would have progressed faster. So we've understood where we are. We've understood that disease-modifying is not the same thing as life-altering, because it's not like they say, oh my gosh, I feel so much better since I've been taking one of these other drugs. It's more like in time you'll have done better. Now, I have had patients say they do feel better, and it's unclear to me whether it's their particular perception of what they're doing, unique to themselves, perhaps unique to the kind of disease they have. Most of the time, patients say they don't, and perhaps you can address this when we have a conversation, but they don't say, I can actually feel a difference, but we offer them because they're disease modifying. So these discoveries, these approvals have led us to an appreciation of the difference between disease modifying and actually symptom changing or life altering. The other thing, which is sort of a humbling, almost tongue-in-cheek reality, is we've become tolerant of negative trials. I mean, in my, as I say, 30 some years as an investigator in studying or helping patients with motor neuron diseases, we're kind of used to a negative trial. It's not a good thing to be used to a negative trial, but in the beginning, when you put all the effort in, you do a trial and you get the result back, it's negative, you're quite despondent. And here you're like, oh, geez, another negative trial. It's become so frequent. And it does raise the question, has our bar for what's acceptable in terms of efficacy or benefit, have we lowered the bar? I don't know the answer to this, but I'll tell you that the drugs we have now did change the slope, did show statistical significance. And for those reasons, the FDA had valid reasons to approve them. But I wouldn't call them a home run. I wouldn't say we have something that's radically changing the disease, but we've accepted them because we're just glad to have drugs that affect. And I think in many areas of medicine, we started out with minor discoveries. The first HIV drugs, for example, were not nearly as effective as the HIV treatments that we have today. And I could say the same thing for cancer and many chronic illnesses that had to start somewhere. So we're all hopeful that the door is open with these disease-modifying therapies, and we haven't necessarily seen the last word on this. The other thing that these trials and our understanding currently has given us is an appreciation for the heterogeneity of the disease. And this is a topic that's of personal interest to me, and it's my own research focus. I personally, and I'm not speaking for the field in general here, but I really think that it's time to think of ALS not as a disease. It's time to think of ALS as a spectrum of disease, and no longer do we assume that everybody has exactly the same. We see heterogeneity at every aspect of this disease. We see it clinically, and I'm sure you're even more aware of that than I am in terms of how what you're going through might not be what somebody else on this call is going through. We see it genetics. There are many different genetic triggers or genetic problems that can trigger a motor neuron decline. We see it in the physiology. The fact that there's 8 to 10 to 12 different hypotheses, that's heterogeneous. There is variability everywhere you look at ALS. And that variability has been thought to be sort of a confound, meaning it kind of gets in the way of the interpretation. And some of the trials that we've done in the past have tried to lessen that heterogeneity by enrolling a more selected group of patients to hopefully better understand that it's helping at least one group and the heterogeneity isn't diluting out something. But heterogeneity and understanding that heterogeneity is something that we have become quite adept at. And then cohort enrichment is the phenomenon I just mentioned, where you kind of purify the enrollment group by enrolling patients that are perhaps progressing at a more advanced phase or a less advanced phase or have a certain stage of the disease. That's actually how Edaravone, Radicava, got approved. Because the third trial, which turned out to be the positive trial, was different than the first and second trial because the enrollment group was different. They chose a much more selected small subset of the total population, and that's where they found the difference. There might be a lesson there. Hold on a second, let me move this out. So the understanding of disease heterogeneity, the clinical heterogeneity, has to do with understanding the disease. ALS as a motor neuron disease involves pathology of the upper motor neuron, that's the motor nerve cells in the brain, the lower motor neuron, that's the nerve cells in the spine, and some degree of progression. And this overlap between the spectrum of upper pathology, upper motor pathology, and the spectrum of lower pathology is what we call ALS. But in fact, as I've been telling you, this overlap is quite variable, and maybe so variable that it is more than one disease. The question is, we don't exactly know how to slice the pie, which variables make the most difference. Perhaps if somebody has upper motor neuron predominant disease versus lower motor neuron predominant disease, should we be calling them different? Should we be studying them differently, or should we be clustering everybody? Maybe it's those that have the frontotemporal dementia that we should be separating out from those that don't have the frontotemporal dementia as an important covariate to lessen the heterogeneity and more or less purify or focus on the group. Maybe it's symmetry. Some patients have a symmetric bilateral presentation. Some are more asymmetric. Some are earlier or later. Maybe it's whether you have bulbar involvement or not bulbar involvement. So this Venn diagram, this thing that I'm showing you, understandably gets very complicated because we don't actually know yet how to slice the pie with respect to the heterogeneity of the clinical presentation. At the moment, clinical trials today, you get enrolled if you meet the diagnostic definition of ALS. There's lots of variability in that diagnostic definition. And we don't know if we should be better studying bulbar patients compared to limb patients, or upper motor patients compared to lower patients, or those who progress fast or slow, or those who have the frontal temporal dementia or don't, localized versus generalized, and so on. So it's a very active area of not just my research, but others as well, in trying to figure out, if I can use the euphemism, how to slice the pie to figure out which is the best subgroups to test if the clinical heterogeneity is one of the reasons we haven't seen a more robust clinical response. Now, changing tone a little bit, I'll say one of the most exciting advances is our understanding of the genetics. Now, this graph is a little complicated to understand, but each of those letters, numbers, and labels are different gene mutations that have been discovered over time. And the line, the black line, shows you the rate of discovery. The size of the circle reflects how many of the patient population might be affected by that mutation. So the SOD mutation, which I mentioned earlier on, was the first of our understanding of the genetic abnormalities that can underlie motor neuron disease. That's about 15 to 20% of the total population. But this big one here, the C9, back in the middle of the graph here, C9 can account for 40 to 45% of the total population might have that mutation. So understanding which genes are involved in which patients might be an important opportunity to look at patients differently, look at different patients in different ways. And these different gene abnormalities are responsible sometimes, I'll show you in a moment, for different cellular mechanisms. So it kind of makes full circle that now we have some genetic markers that underlie some of these proposed pathologies that could give rise. And this is what I was referring to. So, for example, impaired protein homeostasis, that is addressed by a variety of different gene mutations. Neuroinflammation is addressed by some other mutations. Now, it's not so clean that it's only one gene, one mechanism, but it gives you an idea that maybe if we paired our understanding of these different mechanisms and we paired our understanding of the different actions of some of these mutations, we might get a better idea of how best to address this heterogeneity. So the yellow boxes are where the SOD1 protein is active, in which mechanisms, and the red boxes are where the C9orf72 mutation is active. I chose these two because they account for quite a bit of the population of even non-familial ALS. And so it gives us an understanding, a little bit of a vantage to look at different mechanisms, perhaps with different genetic markers. Stop advancing here. Okay. Now, I don't mean to confuse you, but I'm basically introducing the same idea with a different slide. But these cellular processes of oxidative stress, of calcium dysregulation, of difficulty with moving objects, moving subcellular organelles within the motor neurons, they're not independent. Sometimes one pathology can give rise to another pathology, can ultimately result in cell death. And the stars that you see here, this is the site of action. So this is where riluzole is active, up here at limiting the glutamate's effect onto the motor neurons. This over here is thought to be where Relyvrio is active, because we think it stabilizes the mitochondria as well as the endoplasmic reticulum. And that's blocking this cascade of events that lead to cell death at this location. And this here is where Radicava is affected in limiting oxidative stress. So the idea that these individual processes, whether they're triggered by genetics or triggered by the environment, they're kind of related. So one pathology can lead to another pathology, can lead to another process, which can result in cell death. So one thought is the more places you interrupt this cascade, again, by taking multiple medications, perhaps that's a better way to more ultimately create a bigger difference with regard to limiting or lessening the degree of cell death. So we have a very heterogeneous population, and we're struggling to try to figure out how to segregate it. Do you segregate it by, as I say, the frontal temporal patients? Do you segregate it by who's bulbar versus limb? Do you segregate it by who is progressing quickly or who is progressing slower? Many attempts are going on at the same time trying to figure this out. And then, of course, there's a whole area of biomarkers looking at biochemical mechanisms that could reflect disease activity and try to figure out which drugs have a bigger impact on which groups. This is basically the same thing as I've been saying. I'm going to skip over that for just a moment. This was an interesting graph, and it's relevant because it is part of a review, and it shows how the degree of motor neuron death, that is the number of motor neurons that are involved, are understandably very related to the number of problems a person has. So on the x-axis here, it's the degree of upper motor neuron loss. So if you have 100% upper motor neuron loss, you have perhaps this amount of disease severity and this amount of motor neurons affected. If you have a mix of upper and lower motor neuron in the middle, you have more motor neurons affected than if it was just lower motor by itself. And if you have upper and lower together, and you also have frontotemporal dementia here, you have a whole nother set of cells involved. So it gives you an idea that the phenotype, the presentation of the disease is definitely correlated to the number of motor nerve cells that are implicated in producing that. Not a surprising, but I like the way it looked graphically. It also talks about there's a whole lot of patients that fit in between these points, and they're not all in discrete clusters. I'm going to skip this. So I want to move to the last section before we open this up for discussion, which I'm looking forward to, and to say, where are we going? I've highlighted for you a number of discoveries. I've highlighted for you the rate of the progression and some of the problems that we have. So there are a lot of drugs in the pipeline, a lot, more than I have time or understanding to talk about in detail. But we want to make sure that we are fully appreciating the lessons of the past. And what we've got recently is drugs like, so here's a table that I took from a friend of mine, Dr. Andrews, also at Columbia, showing just the number and diversity of drugs that are in the pipeline and some that have already been tested and approved. And we have new ways to test, like the platform trial. Those of you who are involved in the platform trial, and we're participating in that as well here, the platform trial is a very efficient way to test a large number of drugs because we get answers much faster. But the reason it's efficient is because we're doing the same protocol, the same testing mechanisms on each drug in succession, and oftentimes at the same time. It's a great idea if the protocol that we're testing is measuring what we hope to measure. But if we're missing something, we're going to miss it a lot of times. So what might be very efficient, we hope is sensitive. But it is one of the concerns about putting all of the eggs in one protocol, so to speak, all of the eggs in one basket, all of the eggs in one protocol in hopes that we pick it up. But it is so far been highly efficient. We're on our seventh drug in the platform trial in a fairly short period of time. It would have taken us many more years to get through that number of drugs in the traditional model that some of these others have gone through. But realize that there are a lot of different drugs, perhaps more than ever, in the pipeline that need to be tested to see which one has benefit. But at the same time as we need to test them, we need to think about how we test them. And are we testing too heterogeneous of a group, or is that the right approach? I'll tell you briefly about one initiative that I'm involved with here, and we've come up with a scale that will basically label or stratify patients who have different presentations. The scale addresses the extent to which you have upper motor neuron disease versus lower motor neuron disease versus bulbar disease versus cognitive disease. That is the signs of dementia. And we stratify by identifying different indexes, A, B, C, D, based on the severity in each of these categories. And we look to see if you present with one category, do you migrate to another, or do you continue on in that same category? Should we be stratifying in clinical trials? We're still working on this, but this is one of our attempts to try to understand better and follow better the clinical heterogeneity of the population. I'll tell you, we just recently, just this month, did our first analyses looking at these different groups on patients that were taking edaravone, Radicava. And we found actually that not every group, not every subgroup that we identified responded the same to Radicava. Some groups responded better and some less. It's not a definitive result yet because it was an interim analysis. We didn't have all of our numbers in yet. But if this holds true, if what we have initially found holds true, then we might have a better understanding of who should be getting that drug. Perhaps it's not equally beneficial for all types or presentations of motor neuron disease. So this is a line of investigation just to give you an idea of one of the ways to study. We're studying oxidative enzymes in different subpopulations of ALS. Now, some of you I know are interested in cell therapies, stem cell therapies, and some are gene-targeted therapies, quite a pipeline there as well. And some of the very exciting data coming out of the gene-targeted therapies, which I'll talk about in, I think, the next slide. But the cell therapies are also very popular. I know that there's a lot of work going on right now as to decide what comes next in the stem cell group. I think it's a very valuable area of research. It's very interesting. I'm curious to see where this is going to go. But again, a completely different approach than what had been tried before. Now, the last thing I'm going to introduce is this idea of personalized approaches, and it came out of the gene therapy or targeted gene therapy approaches. I think this past year, the most exciting talk that I have heard in this area has been from the group in Columbia. Dr. Neil Schneider gave a wonderful presentation at a meeting that I was at, and it was about his program called Silence ALS. And although it doesn't apply to a lot of patients, what I saw made so much sense to me. And what he did was he took certain patients that had very specific mutations, and he developed a drug, if you will, a small molecule drug, if you will, that silenced that mutation for that patient. So he didn't generalize it to a group. He generalized it to that mutation in that person. And he presented some very profound data about how the disease was actually arrested in a condition, in one of the forms of ALS, at least in that particular individual. If this holds true, and it can be generalized, then knowing whether you do or don't have a specific genetic abnormality, maybe that will be a very important advance in understanding which personalized medicine to silence your particular mutation would be relevant, not necessarily for your neighbor or your friend, but for you personally, and then the next person gets sequenced differently. So it was very interesting. I wish I could have shown you more of his data, but I didn't get permission to share the slides of the actual a change in the clinical course. But there were, I think, one or two sets of twins that they had the identical mutation. One did quite poorly, and one did very well with, or better with the specific targeted therapy. The other approach, which we're hearing more about, and I also think is quite fascinating, and I've been involved with for a while myself, has been combination drug therapies. And that is, should we be doing trials with single drugs anymore? Or should we be combining these drugs? Should we be looking and saying, like we did with the sodium phenylbutyrate and TUDCA, should we be combining all the time? Should that be our new norm instead of doing single drugs at a time. There's another initiative from a small company in Israel that has a combination therapy that's being developed right now in other countries, and we're hoping to be able to do that trial here in the US. But it too is a combination therapy. A lot of reasons, especially with multiple mechanisms and a heterogeneous group, that a combination of a drug therapy or what we call cocktail therapy might be a way to go. This is a symposium that I was actually fortunate enough to organize 23 years ago in 2000 it was a consensus conference on multi-drug therapy. Well, for most of those 23 years, we haven't really talked very much about combination drug therapies until recently. And now the fact that it's becoming more of a everyday type topic in the community is very gratifying. So again, we have multiple mechanisms, we have multiple ways of addressing those multiple mechanisms, and this is a very busy and active area of research. I'm going to skip on the biomarkers, and I just will wrap this up to say that I think it's time to embrace this heterogeneity, and that is to say, we shouldn't just talk about it as a problem or a confound in our trials. We should embrace it. We should study it. We should stratify our data and see if certain patients respond differently than others, and not necessarily accept every patient as necessarily having the same diagnosis. Perhaps same diagnosis, but different reasons, different medicines. And this idea of a personalized approach, whether it's the genetic mechanism that I mentioned, or it's a biochemical drug, in the future, my hope is that a new patient will have an assay done that will be developed that will say which approach to take for that individual, which might be the best approach, best therapy, best medication for that. And maybe we won't have to show this slide in the future. Maybe we'll understand much better which is the way to cut the pie by this heterogeneity. And it won't be a problem, but it'll be the actual vantage in to better understanding of the disease. Perhaps it'll be the scale that we're coming up with here, or maybe this isn't the best idea and we'll come up with something in the future. But we've got to start to look at why did so many good ideas for drug discovery wind up in failed trials? Could they all be wrong? Could all of those data that led to those very well-designed clinical trials all be wrong? Possible? I don't think likely. So hopefully I've been able to give you a sort of a whirlwind snapshot of where we've been in ALS research, where we are right now, and then where we're going, and also giving you a little bit of a look behind the curtain at some of the things that we think about as to why we haven't made more dramatic, if you will, discoveries, and where we're hoping to make a difference in just a very short time. So I really appreciate your attention. Hopefully that was of value to you, and I'm very much interested to hearing your thoughts and questions. Thank you.
Thank you, Dr. Rosenfeld. And we're going to have Q&A, and my colleagues here who are student fellows, Casey and Brian, will moderate the Q&A. Casey and Brian, take it away.
Hi, Dr. Rosenfeld. Thank you so much for the very informative presentation. Our first question is, how is the research community employing AI in the search for commonalities in therapeutic treatments?
Yeah, it's a good question. And I have to say, I'm sure there probably are people that are working on that, but I'm not aware of a particular AI approach to understanding the dilemma, some of the dilemmas that we're in. And not to dismiss this, but part of it is I'm not sure we know what to ask AI to do it. I mean, do we say, how best, AI is as good as the information that it's using to come up with the answers, and we don't have that information yet. So I think we are going to see the benefit of AI as we understand better some of these individualized approaches to subsets of the disease. Then it may make a lot of sense. But at the moment, there might be, but I'm not aware of a research effort that specifically uses AI.
Perfect. Thank you, Dr. Rosenfeld. The next question is, why hasn't the European community approved Relvio?
I'm sorry, can you say that again?
I'm sorry about that. My question is, why hasn't the European community approved Relvio?
Relivrio.
Relivrio, sorry about that.
Yeah, that's what it was. So Relivrio is the sodium phenylbutyrate TUDCA, and it was not approved by the European authorities just this past week, I think. I didn't hear the hearings, I didn't hear the details of it, and so I don't know the answer directly, but I'll tell you that I suspect the European regulatory agency is quite conservative, and some say it's more conservative than the FDA. There's currently a phase 3 trial for sodium phenylbutyrate and TUDCA. It's ongoing right now, and it will result, I don't exactly know when, but sometime soon, within the next year, I expect. And it is the definitive phase 3 trial. And I suspect that the European authorities wanted to see that result before they approved it. And they did not want to approve it based on the phase 2/3 results that the FDA approved it on. And if the drug's benefit holds up in that ongoing trial right now, I would expect that they would approve it. It might have just been too early for them.
Our next question is sodium phenylbutyrate antioxidant?
It's not thought to be an antioxidant. Sodium phenylbutyrate, and to be truthful, they're not really sure why it works, but it seems to stabilize different parts of a cell. It seems to stabilize the mitochondria, which is the energy machine of the cell, and it seems to the drug Relivrio seems to stabilize the endoplasmic reticulum. But the evidence that is exactly the benefit of Relivrio is not solid enough yet. So in fact, I think the FDA has sort of frowned upon the company making the claim that that's why the drug works, because the mechanism is just not well enough understood. But that's what's implied that it's doing. It's stabilizing the organelles. Of the 3 medicines that are approved currently, Radicava is the antioxidant.
Okay, perfect. Thank you very much. Next question is, has there been any research done concerning viral or bacterial infections that could possibly be a trigger for motor neuron disease at some point in our life?
Yes, actually, there's been a lot about viruses. And I'm working with a small biotech company in San Francisco that believes that the way that the disease propagates, and in their case, they have a small molecule to address it, is the same mechanisms that a virus uses to take on a host cell. And the problem with that area of research has been that we haven't found a consistent virus in enough patients to say that's the cause. But could it theoretically be? Theoretically, yes. There are viruses that we know about that cause motor neuron disease. Polio is a virus that causes motor neuron disease. It causes polio. West Nile virus is a virus that can affect lower motor neurons, but they don't give you ALS. They give you, sometimes generalized, sometimes local motor neuron. But patients that have ALS don't have West Nile virus and they don't have polio virus. But theoretically, could they have a virus we haven't found yet? Theoretically, yes.
We have another AI question. Could AI interpret data to account for heterogeneity-driven variables if we revisited old data to discover whether failed therapies actually succeeded for subsets?
Yeah, that's a complicated question because, of course, I can't say never, but theoretically. But again, what you put into AI is directly relating to what you get out of AI. So if we put in all of the data that we had on a particular large clinical trial, and we put in the patient presentations, and we put in their breathing capacities, and we put in their functional rating scores, and we put in whether they were getting the drug or not, if that was possible, could we be, I guess the question is, could we have been missing something in the traditional analyses? Theoretically, yes, but some of these trials, especially the bigger ones, they were quite extensively analyzed. And I suspect if we were missing something, it's because we didn't measure it. We were missing it because we didn't measure the subgroup well enough to define it. And if we didn't define in the clinical trial or capture the data to say this person is really different than this person, then how can I expect AI to find it if we didn't measure the thing that would allow AI to find it? So I don't know. I guess theoretically, the answer is I hope that is true, but I don't know how that would be the case right now. Maybe I need to know more about AI, but I'm not sure about that.
All right, perfect. Next question is, if we had a complete ALS census with each person's place, history, and the genetic profile, would this provide data to narrow down the causes of ALS? And for a follow-up, could this lead to better understanding of the environmental factors with a specific genetic makeup that could increase the risk of ALS, or I guess any other motor neuron diseases?
Well, sure, more is better, and there's a lot of effort. In fact, we just submitted I was part of a grant that was submitted this week looking at better capture of more patients with regard to their demographics and hopefully their genetic background. But yes, so it is still possible that there are very important biological environmental triggers that could be identified in clusters. But I'll tell you, as an extreme example, there was a time not that long ago that there was a particular cluster of ALS patients at a particular military base in one state. And when it was looked at, it was very interesting because they found that many of those patients actually had exposure to the same building on that base in that state. And I believe they even found that most of the people had exposure to the same floor of that building in that base in that state. And all of that investigation, all of that promise did not really lead to a specific causative trigger. There's been other endemic areas of ALS, and sometimes it's thought to be a fungus in the water, and sometimes it's implicated to be a virus, and sometimes it's implicated to be a mold. But the truth is, we haven't found that yet. So hoping that we would find it with more examples, sure, I think it's reasonable. It's definitely a valid approach. But even when it looked like we were onto something, it didn't result in, a home run.
Our next question is, what are the names of gene panels for ALS?
They don't have names, but they have companies that support them. So for example, today, I believe that there are 2 companies that actually have gene panels that are free to you. That is, they'll run, and I have been sending every one of my patients through these. I didn't used to do this, but I do now. I send every one of my patients that is willing to get this gene panel. So there's a company in San Francisco called Invitae. And Invitae has a panel that's got a lot of the ALS genes, and they can do the test by a saliva sample. So in some cases, you might even get the kit sent to your home. You fill up the little tube that they give you with saliva and then send it back to them, and they'll do the analysis. The other company that is very popular is, I think, Prevention Genetics. Prevention Genetics is a different company, and they have a different panel, and I believe it is also not cost to the patient. Interestingly, they don't measure the same genes. I think Prevention has a few genes that Invitae does not have, and there might be vice versa as well. So we're working that out as to which is the best gene panel. But those are the 2 companies that I'm aware of that have free programs currently.
Perfect. Thank you very much. The next question is, how is ALS therapy effective when diagnosis is very late? And for example, for this question, an average of 11 months or more.
If I'm understanding the question, you're asking, is therapy, is there a point at which therapy couldn't benefit you because you've been too advanced?
Correct.
Is that a fair assessment? Yeah. So obviously having more motor neurons alive is better. It's kind of like if you were going to fertilize the grass in the front of your house, it's better if it's green when you start, right? If you fertilize brown grass, it's less likely you're going to get a green lawn. If you start with greenish grass, you're better off there. It's the same thing. More is better. Although the question specifically said 11 months, it's not quite so simple, because if you slow down the progression, let's just say an extreme, if you stop the progression in its tracks and you have only 20% of the normal number of motor nerve cells at that point when you stop the progression, you're quite clinically affected. But if truly there was no degeneration past that point that you stopped it, the 20% of the cells that survived might be able to take over some function for some of those 80% that died. In fact, in polio, that's what happened. When kids, especially kids, were paralyzed in the 50s with polio, or in the 40s and the 50s with polio, and then the polio was arrested, the virus was arrested, why did they walk again? They didn't grow new motor neurons. They had the healthy motor neurons, or the surviving motor neurons, take over the effect. So that's a bit of a abstract reference, but I don't ever say that there's nothing we can do. And I am always hopeful that there's something. I think there is a hopeful alternative at every stage of disease progression. And I believe that. I believe it may not be a drug therapy. It may not be that I can put those motor neurons back, but I cannot believe that there isn't something that me or one of my team can do to make life better, regardless of how advanced you are. So your question that you asked is specific to whether drug therapy would be at a point where it's too far gone. I guess that depends on the particular drug therapy. But I do think if we could find some way to arrest the disease, there wouldn't be a point at which you couldn't find anything to rescue.
Thank you for answering. Why do all drug studies disqualify people with ALS with over 2 years since diagnosis? Would it be beneficial to include long-term survivors of ALS in drug studies?
Yeah, so that's a very good question. It's a common question. And I would have to say it's a bit of a practical question. So we talked about the heterogeneity. If a disease has been going on 5 years, in other words, slowly progressive, the drug company that sponsors a trial, they want to find an answer, but they don't have years to study it. They don't have the money. It's so expensive. They need to see it in 6 months. But if you're progressing ever so slowly because of the fortunate nature of the progression, 6 months is too short. So what the reason for them limiting the disease duration is because chronic slow progressing patients would dilute out the ability to find an answer. It's not that they don't think that drug could help a chronically slow progressive patient. In fact, it might help them more. It's that we can't find the answer in 6 months if you're progressing ever so slowly. And that's an unfortunate reality of what decisions we have to make when we look on. And interesting to your point, I've actually proposed trials that will look at only very rapid patients and compare them to very slow patients. And perhaps we'll be able to do that with this latest proposal, but it's very challenging to study slow progressive patients over a short period of time.
Perfect. Thank you, Dr. Rosenfeld. So this is the last question of this meeting since we're running short in time. So last question is, could you please explain increased tone and spasticity?
Wow. I think I need a whole nother session. So I'll give you the quick term for that. So as I mentioned, there's two kinds of motor neurons. There's the motor nerve cells that live in your brain and the motor nerve cells that live in your spine, upper versus lower. The ones in the brain communicate with the ones in the spine, and the ones in the spine communicate to the muscle. They have different jobs. The ones in the brain tell the ones in the spine what to do, and the motor nerve cells in the spine tell the muscle what to do. Now, if the brain motor nerve cell stops working, if the upper motor neuron stops working, the spine motor neuron may be perfectly willing, able, and healthy to do something, but it gets no instructions. So there's nothing to tell it how to make a movement. All it knows how to do is fire. And so it fires without the regulation of the brain motor cell. And that tends to be a very stiff and spastic movement. So it's the absence of the controlling influence of the upper motor cell onto the lower motor cell that gives you increased tone and spasticity. It is an upper motor neuron problem. And some patients have only upper motor neuron problems. That is, they're not weak, but they can't use the muscles because the brain isn't telling the spine how to use the muscles. And without the brain's influence, the cells are very active and they're firing all the time and you're stiff and spastic.
Perfect. Thank you, Dr. Rosenfeld, for your amazing presentation. And that concludes our Q&A portion of this meeting. I would like to send this back to Indu for any final words, remarks, concerns, etc.
Well, thank you very much for your presentation, Dr. Rosenfeld, and this was excellent session. And we move on to our open forum from here. And you're welcome to stay, but I understand we took a lot of your time today, and we are very grateful for all the hard work you do to bring therapies for people here. And we're really grateful for all the efforts you put in. And we'll open up and we'll stop recording and we'll open up to Q&A. But if you have any, before I hand it over, if you have any last tidbit of what you think about our community and what was your experience like tonight, we would love to hear it.
Well, I mean, I'll just say briefly, it's really, as I said, it's a pleasure to be here. It's very impressive that so many folks come from such a wide spectrum of the country, and I guess even out of the country, from what I'm seeing. I'm looking, as you're speaking, I'm looking at the chats. We didn't address a lot of questions. I feel bad about this. I could have talked less and answered more questions. I didn't realize there'd be this many. If you have a burning question that you really would like to have some comment on, feel free to send me an email. You can either send it to me at Loma Linda or send it through Everything ALS and they'll forward it to me. And I'd be happy to try to address it as long as it's addressable in an email. I'll be happy to do that. But it's really been a pleasure. And again, I just want to thank you so much for the opportunity.
Thank you. And we will definitely, share the questions that we get. So thank you.
Absolutely. Thank you very much.
Thank you.